everyone. Uh, I'm going to present my tool, hkgfinder. It's a tool that can help you find and classify housekeeping gene from genomic and metagenomic data. Uh, I hope you are all excited as I am to be here today. As you can see, I come from Côte d'Ivoire, but just before we can dive into the tool, we will set up the background. The first question you should ask yourself is, how do you assess microbial diversity? And this picture summed up somewhat in how you can do it. You just have first to sample a microbial community, and second, before you two roads. You can go for metagenomic sequencing. When you choose this road, you have all your DNA that you are going to sequence. But when you choose to go for amplicon sequencing, you have to choose one gene to target. And the number one gene targeted in amplicon sequencing is cysteine sRNA gene. However, this domination comes from there's two papers from Carl Voss Lab in late 70s that established the cysteine sRNAs for phylogenetic analysis of prokaryotic sequences. But as you can see, even today in mGenify, most of the studies are still single gene based. However, we know that cysteine sRNA are limited, and the hypervariable region are also limited for taxonomic resolution of some genera. And this picture shows a study we did for rhizobia, which are a type of bacterial that have a symbiotic relationship with plants. And you can see that for the 18 genera of rhizobia, all the major hypervariable region we are using commonly are not able to resolve the different genera of rhizobia. You will have to take the full 16 sRNA. And even in some genera, the cysteine sRNA is not able to taxonomically resolve the genera. Here we are going for housekeeping gene, and some studies have already demonstrated that housekeeping gene can help in such station. So we have the first studies that show that RPOB is a promising gene for such studies, and we have also some more studies and multilocus sequence analysis. This is why, but how you can find housekeeping gene in genomic or metagenomic dataset? You have genome annotators. But if you go for genome annotators are going to classify all genes. So they will take more times, and when you have the data, you have also to filter out the gene you want, and that is not so easy. This is why I developed hkgfinder, and its novel value lies in its ability to directly predict the housekeeping gene you want, and at a high specificity and sensitivity, and faster than genome and metagenome annotators. hkgfinder is based on hidden Markov profiles, and I built it by taking seed alignment that we just built using sequences, and also seed alignment from PFAM and TigerFAM. And we built 18 hidden Markov profiles for 70 housekeeping gene. Among others, you can see RPOB and DNAK, and a picture of some of the models. How hkgfinder works. So you have three modes. You have the normal mode, you have a faster sequences, and if it's DNA, it is then translated in proteins and just search against the HMM profile using HMM search. If you have a genome, you can just select the genome mode, and then we will run prodigal with the correct option, and also the metagenome mode with the correct option, and then you can classify using HMM search. I've implemented hkgfinder in Python 3, and Python 3 version 3.8 to 3.11, and hkgfinder is parallelizable due to the amazing Cython packages. I use PyroGigal and phmember. I think Martin is the room. Thank you, Martin, for developing phmember and PyroGigal that help us to parallelize the tool. And because we use this two tool, we don't need external dependencies to run hkgfinder. You can just install it, and you don't have to install hmember or PyroGigal. When you run hkgfinder, you have the main output is a text file that output the name of the sequence and the predicted gene and the e-value and the bit score. But you can also get an annotated FASTA file with the gene and the family predicted. We did some evaluation of hkgfinder. It's difficult to do, but it's just difficult to do it using genome annotators to compare with genome annotators, because in the back they use the same tools. They use Markov profile, so we are going to expect the same result as hkgfinder. So we just compare it against BLASTP, and we obtain that in average, hkgfinder is 219 times faster than BLASTP. And you see that hkgfinder has a great sensitivity and specificity. The gap we've seen is because we include in the test data, in the data set, some partial sequences for which hkgfinder is not so good, but it's probably due because of the fact that hkgfinder run HMM search using the gathering cutoff. We don't use the e-value cutoff. I would like to thank my team in Côte d'Ivoire at Institut National Polytechnique, my advisors Dominique Kwa and Adolphe Zizé, and I would like also to thank the ISCB and the Function COSI and the chairs of the Function COSI that helped me a lot, and for the travel grant who helped me to be here. Take home message: if you want to do amplicon sequencing, check housekeeping gene. Don't rely directly on 16-as RNA. hkgfinder will help you to find housekeeping gene in your genomic or metagenomic data. And last but not the least, I'm looking for a postdoc. I fluently code in Rust for 2 years now. I code in R, I code in Python, I code in Perl, everything you want. I run the Rust for bioinformatics Twitter. I don't know if it's now. Community with Rob Petro, who is not here. So it's me, Bian. And I'm hyper motivated and work for free if you give the food and the house. Thank you.
Thank you. So if you have a postdoc position open in the lab, please seriously consider him. Questions for Anicet?
Thanks for the talk. So the issue about the thresholds, the gathering thresholds. So is that because you're using a TigerFam family, and so TigerFams has very high gathering thresholds? So is it just sort of a TigerFam that's causing you the problem?
Yes, but you can use the gathering cutoff, or you can just use compete the different e-values you obtain when you run the HMM search. We just when we were developing the tool, we just go for the gathering cutoff, but I'm exploring the idea of testing against using others option to see what is the best option. But I think it's a good idea, and yes, it's the gathering cutoff that did it. But the gathering cutoff gave some confidence in giving a good recall.
Okay, and I guess some of those housekeeping genes,
Yes.
the models are in clans, PFAM clans, and so they do hit other related proteins. So I
Yes.
guess those you need to take care.
Yes. Thank you.